All right, so thank you very much for the organizers for setting this wonderful webinar series up. And thank you for the invitation to give this talk. And thanks for all of you who joined this webinar. So today I'll talk about DBS in dystonia, and I'll talk about this topic from the brain network perspective, informed by causal brain lesions and multimodal neuroimaging in patients with DBS. I divided the talk into 3 different sections. So first I'll give a brief introduction to dystonia and explain why brain lesions and imaging in patients with DBS could provide valuable information about the network perspective in effects of DBS. Next, I'll go in more deep into the use of causal brain lesions and how to use these lesions to study the networks underlying different symptoms and specifically dystonia. And finally, about the neuroimaging studies in DBS. And here you can see my disclosures. So I don't have any patents of any of the methods presented here, and nothing that's relevant for the content of this talk. So a historical note. So as you probably are aware, dystonia was not always considered a neurological disorder with background in brain pathophysiology. So at the early days, dystonia was considered a mental health issue with psychological mechanisms behind it. But of course, this view changed, and I took this figure from a relatively recent review highlighting some of the major milestones in dystonia research towards our current understanding of dystonia. And I took the liberty of highlighting a couple of events in this timeline, which are relevant for this talk. So early on, lesion studies in animals were highly influential in reformatting the idea of the background of dystonia towards brain pathology. So creating lesions in animals, researchers were able to induce dystonia in these animals. In addition, there were human case reports of lesions causing dystonia. And also reports of efficacious pallidotomy really nailed down that we are dealing with a brain disorder here. And these early findings with multiple phases in between eventually led to validation of GPI-DBS, which is currently the most efficacious treatment method for dystonia. A couple of words about the disorder itself. So dystonia is defined clinically, and we don't really have any reliable biomarkers that could be used for the diagnosis of this movement disorder. So dystonia characterized by sustained or intermittent muscle contractions that cause abnormal, often repetitive movements, postures, or both. And these movements are typically patterned, twisting, and may be associated with tremor. And dystonia characteristically is usually initiated or worsened by voluntary action and associated with overflow muscle activation. So that's the definition of dystonia, and the diagnosis is clinical. And there are also some other interesting clinical phenomena, such as the sensory trick, which means that the patients can alleviate their symptoms for short term, even quite dramatically, for example, by touching their face in a case of cervical dystonia. Dystonia is currently classified based on 2 axes. So first one is the clinical characteristics, including age of onset, body distribution, temporal pattern, and associated features. And the second axis focuses on etiology, what is in the background of these symptoms, first of all, looking at whether there's any evident nervous system pathology, degeneration, lesions, or nothing. And further, it can be split to inherited, acquired, and idiopathic. So we have a huge variety in individuals with dystonia, with multiple different presentations, clinical characteristics, and also etiologies. Many of the research studies focus on idiopathic, sporadic, isolated cases of dystonia. And that's also the focus of my talk, except for the use of lesion-induced cases to inform us about mechanisms of dystonia in general. However, I want to highlight that dystonia is not stable. So when the patient is initially fit into these diagnostic categories, that may change over time. So dystonia may evolve along with the disease course. And for example, distribution of dystonia can change. It can spread to other body regions compared to which region it started from. So indicating that these dystonia types, at least in terms of body distribution, are not separate diseases. They are clearly linked and part of the same continuum. But still, there are also differences between different dystonia types, even in terms of which part of the body is affected. So some dystonias tend to spread more often than the others. Some are more often associated with sensory trick. Some are characterized more by positive ethanol response and so forth. So ultimately, it's unclear whether all dystonias have exact same mechanisms as would be predicted based on the phenomenology, or whether we have a collection of multiple related mechanisms causing different types of dystonia. The traditional models of dystonia emphasize the role of the basal ganglia. And to a large extent, these models have been influenced by findings with lesions. So lesions involving the basal ganglia are typical for cases with lesion-induced dystonia. And these findings fit quite well with the simplified traditional model of the corticobasal ganglia circuits, which include both excitatory and inhibitory circuits that loop through the GPI, which inhibits the thalamus, and the thalamic activity excites the cortex, increasing movement. So according to this model, if GPI is destroyed, you get net excitation to the motor cortex, which leads to hyperkinetic movement disorders such as dystonia. However, the problem with the traditional model, which would be very convenient in terms of figuring out how to plan treatments, is that there are several findings that are inconsistent with the simplified traditional model. For example, brain lesions completely outside the basal ganglia can cause dystonia. Similarly, pallidotomy works for treatment of dystonia, although the simplified model would just suggest that it actually causes dystonia. In addition, many genetic observations have highlighted that there are multiple different brain circuits and brain mechanisms involved, and the pathology doesn't just focus on the basal ganglia. And for example, in neuroimaging studies, have highlighted multiple different brain regions. So we have found abnormalities in many parts of the brain. And here you can see a summary of neuroimaging findings in dystonia, pinpointing the peak neuroimaging finding in each of these studies onto a brain map. And as you can see, the findings are very much scattered in many different brain locations, leaving the anatomical origin of dystonia unclear. So currently, we are moving and have already moved conceptualizing dystonia as a brain network disorder. Here in this figure, again, I'm showing just the highly simplified model of the basal ganglia circuit, but that's far from the entire picture, and there are many other loops and networks that are relevant for dystonia as well. So we know that dystonia is a brain network disorder, but these networks have not yet been fully characterized. So DBS, GBA-DBS, the most efficacious treatment that is available at the moment for dystonia. We identified this target based on observations with pallidotomy, so it was an easy way sort of to find this target. The efficacy has been validated in multiple randomized studies, and it has been shown that many types of dystonia respond to this treatment. The electrodes are, of course, placed in neurosurgical operation, and the first methods were sort of indirect approaches to target the GPI based on distance to anatomical landmarks. And you can imagine that there will be some variability in the exact lead placements when using indirect methods. More recently, direct methods have gained popularity, and here, with the guidance of modern MRI imaging, you can place the lesions in the posterolateral parts of the GPI more precisely, but still there will be some variability between individual patients. The problems we currently face is that not all patients with similar dystonia respond to GBI-DBS, and ultimately the mechanism of action for GBI-DBS is not fully established. Comparing pallidotomies with GBI-DBS, the clinical efficacy looks pretty similar, and that led initially us to think that maybe DBS works as a virtual lesion, but that turned out to be incomplete explanation as well. So moving forward to the next section of my talk. So I'm going to go through observations with brain lesions and how to use brain lesions to figure out which are the critical networks in the brain underlying dystonia. And could these networks, when found out, inform us more about how we should use DBS to treat dystonia? So starting from a historical note again, so brain lesions have been the cornerstone of neurological localization throughout the history of neurology. And even like single cases with striking symptoms can provide a wealth of information how the human brain works and where different symptoms localize. For example, case HM, Henry Molaison, had severe epilepsy, which did not respond to other treatments. He went an operation where both of the medial temporal lobes were resected, so taking out both of the hippocampi. And after the operation, Henry was no longer able to make any long-term memories, informing us about the critical role of hippocampus in human memory functions. You all have heard the story of case Tan, described by Paul Broca, that led Paul Broca to describe the speech production area in the brain that we currently know as Broca's area. So collecting this sort of cases with clear-cut symptoms following brain lesions can be also used in a group level to get more detailed insight on the localization of symptoms. And now if all patients who have lesion-induced a specific symptom, if all of those lesions overlap in one place in the brain, then we can be fairly certain that that symptom originates from damage to that specific spot in the brain. However, the problem is that quite often lesions causing almost identical symptoms do not overlap at all, leaving the localization unclear. And we know that all human brain function is dependent on the intrinsic connections within the brain. So a single neuron connected to nothing does nothing meaningful. You need a network of neurons. And pretty much all meaningful brain functions actually localize to brain networks. And now if in this context you consider lesions, lesion anywhere in a connected network will disrupt the entire network function, leading to symptoms corresponding to that network function. And there are many ways how to study these networks, structural connectivity, looking at anatomical connections, or functional connectivity. Both work pretty much with the same principles for this purpose. And just to give you a very simplified idea how this works, is that we start by collecting a number of cases who have suffered the same symptoms caused by a lesion, then define the connections disrupted by this lesion, resulting into so-called lesion networks. This process is repeated for all patients who have lesion-induced same symptoms, and then we can look for commonalities in these lesion networks. And if there are regions that are shared by all of these networks, that means that every single lesion that caused this symptom was connected to that spot in the brain, indicating that that location in the brain and its connections, a network defined by connectivity to this spot, is likely to be causally related with the symptom. As a final slide before moving into findings in dystonia, which are the key point of this talk, just wanted to show that this approach has been used to multiple different neurological and psychiatric symptoms, and each time it has seemed robust enough to detect networks underlying the symptom. It's a powerful approach because all of these patients have lesion-induced new onset symptoms. And just to give you one example of these conditions. So first, we studied quite recently lesion-induced ataxia. So ataxia that localizes to the cerebellum, right? Everyone knows that when you see a patient with acute onset limb ataxia, think of stroke in the posterior circulation, at least in the adult patients. What we discovered in a sample of 200 consecutive stroke patients was that actually more than half of the lesions causing limb ataxia were localized outside the cerebellum and outside the cerebellar peduncles. However, those lesions were connected to the intermediate zone cerebellum, indicating that it's a network phenomenon, and lesion anywhere in a network connected to the cerebellum in these locations can cause limb ataxia. All right, how about dystonia? So the first study focused on cervical dystonia only, which is the most common isolated adult onset dystonia. Here you can see lesions causing cervical dystonia, and as you can see, there's no common denominator in these lesions. Next, we define the networks disrupted by each of these lesions, looking at the commonalities, and what was discovered was that all of these lesions were connected to regions in the thalamus, in the globus pallidus, in the brainstem, cerebellum, and primary sensory cortex. When comparing these lesion networks to lesion networks from just random strokes and lesions causing other movement disorders, connections that were specific for the cerebellum were the cerebellum and the somatosensory cortex. Now, the key question is that since lesion-induced dystonia is quite rare compared to idiopathic dystonias, And if we think that lesion-induced dystonias can tell us something about the mechanisms in idiopathic dystonias, then the network that was identified based on lesions must be abnormal also in idiopathic conditions. And that is indeed what seemed to be the case. So both of the main network hubs were highly abnormally connected. Basically, the normal connections from these regions to other locations in the brain were disrupted in cervical dystonia patients, indicating that these findings may be relevant across etiologies of cervical dystonia. And finally, to look at whether these networks might have relevance for treatment, the lesion network hubs were compared to efficacious deep brain stimulation in cervical dystonia. So this work leveraged a fantastic study by Martin Reich and Jens Borkman, where they defined the optimal locations of DBS electrodes that were associated with best therapeutic outcomes. So these best locations in the GPI, when looking at their connections elsewhere in the brain, it was noticed that these had similar kind of connectivity to the cerebellum and also to the somatosensory cortex. And there are multiple lines of the same kind of research in other conditions, all suggesting that targeting the causal network might have some clinical benefit. The next question in dystonia networks is that since that study was only in cervical dystonia, does the network perhaps generalize across all different types of dystonia affecting different body regions? So to that end, Daniel Korban and others collected all published cases of lesion-induced dystonias, ending with 359 cases. As described early on, and as the early studies highlighted that the basal ganglia is the typical location of lesions in lesion-induced dystonia, that's also what this synthesis told, but still lesions involving the basal ganglia were present only in a bit less than half of the patients. When comparing the proportion of lesions involving the basal ganglia in lesion-induced dystonia compared to lesion-induced other movement disorders, basal ganglia lesions were clearly more common in cases with dystonia. And the other region that was more common in lesion-induced dystonia was the cerebellum, but only affecting 13% of the patients. And as you can see, there's a quite big spread on where lesions causing dystonia occur. So the next step is to look whether these lesion locations have any relevance in terms of the clinical phenomenology. This was done by using a latent component analysis, looking at whether the data could be split into different classes based on the lesion location and affected body regions. When adding the distribution of dystonia, focal, segmental, generalized, and so forth, to the model, it didn't result in any specific classes. But when adding the exact body regions, which showed dystonia in these patients, the analysis produced three separate classes. These were termed the limb, hand, and cervical class, based on the typical body regions affected. So how you interpret this graph is that if you look at the green line, which represents the hand class, almost 100% of these patients in this class had dystonia affecting the hand. And thalamic lesions were overrepresented in this group. Similarly, with the blue line, there's the cervical class. Lesions affecting the brainstem and cerebellum were most common in this group, and they were characterized by dystonia in the cervical area, eyes, and face. And finally, the the limb class, basal ganglia lesions in approximately 70% of the cases, and these typically had dystonia affecting the limbs. And what is interesting, based on the split with the lesion locations and body regions affected, we also see differences in other clinical factors such as tremor, variability of the symptom latency, and sensory trick, indicating that not all dystonia is the same in terms of neurobiology. So as the next step is then to ask what are the networks then, and how do they differ, and is there a common network in dystonia? So the relevant part of this figure is the lowest row. So this is the network where all lesions causing dystonia were connected to, and it was specific against just connections from random lesions and lesions causing other types of movement disorders. So it seems that there is a shared brain network underlying dystonia, at least based on the information provided by causal brain lesions. Now, if you split the data set into different types of dystonia in terms of the body part affected, you can see that all of these have somewhat similar regions highlighted, regions that were part of the so-called general dystonia network, but they also have clear differences, with different regions weighted more in different types of dystonia. In addition, in the cerebellum, you can see a clear somatotopic organization matching with the somatotopical organization of the cerebellum reported a decade earlier. So different types of dystonia in terms of body regions affected have slightly different network involvement. And finally, when looking at to what degree lesions damage the general dystonia network, the more damage these lesions do, the more body parts are affected. So now to the final part of the talk. So I hope that I managed to convince you that brain lesions can provide us some valuable information about the background of dystonia, and that provided some more evidence that dystonia indeed arises from networks. So how about DBS then? Are networks relevant here? So as you know, it's very efficacious treatment. The efficacy has been validated, but we still have some issues. And of course, the biggest issue is that not all patients respond, even though our targeting seems fine. Because of the targeting techniques and uncertainty in exact lead placement, the exact stimulation sites seem to vary quite a bit between different patients. Again, I'm using the figures from study by Martin Reich as an example. This was a multi-center study collecting a large number of dystonia patients treated with DBS. And what this study actually showed, that lead location does matter, but it's not the whole story, and it's not a simple story. So first of all, looking at the exact sites where the stimulation active electrodes were, and comparing the patients, the distance between the optimal spot and the patient's active stimulation site, they showed that there's quite poor association between the simple distance from the what would be an optimal site. However, in a clever way to look at the data, when considering the whole volume of tissue activated by the active electrode contact, and acknowledging all regions touched by it, a combination of the activation, what they called a VTA atlas model, they were able to explain approximately 50% of the variance in clinical outcome. So it's not a single precise spot in the GBI that needs to be stimulated. It's a combination of locations there, a combination of fibers probably, that would lead to the best results. Other issues that have puzzled us at least are the slow responses in with DBS in dystonia. So as many of you who work with DBS and dystonia know, the clinical benefit appears with a delay. So it may take weeks or even months to reach the optimal therapeutic benefit in these patients. So that's a complication and difficulty in terms of programming. And we recently looked at what happens when you switch off the device. And in this study, there's only 11 patients with long-term DBS, with stable DBS treatment, good response, approximately 70–80% improvement. So in this graph, the 0 represents the preoperative symptom severity in Twister score. Here is postoperative best state. And this is at the time of the study when the patients were on stimulation for years already, showing that they were roughly at their optimal response. When you switch off the stimulation, the symptoms come back almost immediately, but only to a small extent. And when the stimulation was kept off for altogether 2 days, the patients returned to their preoperative symptom severity. And when the stimulation was switched on again, they reached their normal good state with on treatment within 3 days. So it appears that the initial response is very slow, but when you have established already the efficacious treatment, then the responses either switching off or switching on are still slow, but much, much faster compared to when the treatment was started. So all of this indicates that there must be some slow neuroplastic processes, and when these processes have developed, then the changes are faster with modification of the treatment. So what are these slow processes, and how do networks are relevant with these? So this is a study by Andreas Horn and colleagues where they looked at the exact DBS active contacts with the volume of tissue activated models, and looked at whether they could explain the therapeutic response based on connectivity from the stimulation sites. They used an indirect connectomic approach, which is similar to lesion network mapping. So you have a normative connectome built from hundreds of thousands healthy volunteers representing the normal connections in the human brain. And by combining this data with those connectomes, they were able to show that the good response in cervical dystonia was associated with structural connections to the head and neck area in the motor cortex, and in generalized dystonia, good response was associated with more widespread connections to the motor cortex. And the functional connectivity findings spoke to the towards the same idea, highlighting also the same regions that were identified based on the causal brain lesions to be relevant for cervical dystonia, for example. In more direct approaches to figure out what is going on with DBS and do the responses extend beyond the stimulation target. This is a study from Canada where they scanned cervical dystonia patients with DBS. They scanned them with the resting state fMRI, measuring low frequency fluctuations amplitude, which reflects activity in different regions in the brain. They had a clever design where they had optimal contacts, optimal stimulation on, then they modified the stimulation to be suboptimal therapeutically, scanned the patients on suboptimal stimulation, and finally when the patients were off stimulation. And what they found was that there were significant differences in stimulation-induced activation between optimal and non-optimal conditions, highlighting again the somatosensory and somatomotor cortical regions. And using a model with responses in sensory motor cortical regions and SMA, they were able to predict symptom improvement in these patients, showing that the remote activations with DBS seem to be relevant for therapy. There's even a more robust way to find the effects of stimulation using FDG PET. So FDG for deoxyglucose is a glucose analog, reflecting the glucose metabolism in the brain. And this is a very robust but still quite sensitive measure for focal activation in the brain. So in this study, again, 11 patients with cervical dystonia with chronic DBS treatment. The treatment was switched on and off, and brain glucose metabolism was measured using PET. Here you can see the electrodes. You can see a heat map of where the electrodes were most often localized in the GBI as it should be. And what was discovered was that in the GBI, the higher the stimulation amplitude, the more glucose metabolism increased when the stimulation was on compared to off. So in striking contrast to lesions, which basically just destroy neurons, leading to no neuronal activity, in DBS, the activity goes up and it correlates with the strength of the stimulation. However, what was curious is that the level of activation in the stimulation target in the GBI had zero correlation with symptom improvement. It didn't correlate with symptom improvement when comparing the time of on scan and time of off scan, nor did it correlate at all with the overall improvement following DBS treatment. DBS stimulation also induced metabolic activation elsewhere. It was not restricted to the GBI. So you can see that it extended to other regions in the basal ganglia, including the putamen, including the subthalamic nucleus, and sensory motor cortical regions. And I think that especially the STN finding is curious in a sense that there are reports saying that also STN DBS seems to work in a treatment of dystonia. And quite recently, Andreas Horn's team from Harvard looked at connectivity from STN DBS in different disorders. And what they were able to show was that contacts associated with good response in different disorders mapped to different parts of the frontal cortex, indicating again that the networks, exact networks that are stimulated are critical for the response. And what the finding with the GBI-DBS induced STN activation means that STN and GBS are part of probably the same network, explaining why both of these work. And with the network theory, basically there should be other locations as well where you might get some treatment benefit. But back to the PET imaging data. So looking at what correlated with motor symptom improvement, the short-term improvement after switching DBS on was associated with metabolic increase in the sensory motor cortical areas and overall treatment with activation in the SMA area. And when the activity from these clusters was projected back to the GPI to see which contacts were associated with highest activation in these areas, both of these heat maps indicated the same spot, suggesting that the same location that where you can get short-term response is also probably relevant for overall treatment response, at least in this small sample. What is also curious is that GPI DBS is sometimes associated with side effects. So in this sample, worsening of trail-making test B part was associated with stimulation-induced increase in activity in the medial frontal cortex, and activity here projected back to the GPI in a slightly different location compared to where the motor response was obtained. And finally, there was a single case in this sample where stimulation induced Parkinsonism. And interestingly, this patient had the highest of all of these patients' activation bilaterally in the putamen, suggesting that the network effects are relevant not only for motor improvement in dystonia, but also for emergence of side effects. So I realized that I have used pretty much all my time, so moving to summary. So what I hope that you'll get out of this talk is an idea how lesions can be used to inform the networks underlying different symptoms. This approach might be useful across motor symptoms and non-motor symptoms, and each time these seem to map to different networks. Similarly, in the therapeutic side, the efficacy of DBS seems to be mediated based on specific connections from the stimulation site elsewhere, and DBS induces metabolic activations far away from the stimulation target, mediated by the connections of the exact DBS site. So all of this is now at the stage of idea, and I would really hope that I could tell you how to leverage networks and how to place your DBS leads in terms of these networks, but we are not there yet. And what I think that is needed is, of course, step-by-step data, including multiple different approaches and modalities to really nail down where these networks are and how to modulate those for the benefit of the patients. And I'll end my talk here. Thank you very much for your attention, and if you have any questions, I'm happy to try to answer.
How do you choose the target for DBS due to network theory in dystonia? I mean, only GPI or other location? Do you consider dystonia phenotypes?
So currently, I think that, well, we have the best evidence for GPI. So I don't see any reason to select a different target. At the moment, I think that we have data to show that networks are relevant, but we haven't yet figured out how to use the networks and how that affects the exact lead placement. So I'm in the clinical treatment side, I'm quite conservative. So I would go with what is shown by the randomized controlled trials and used standard techniques to target the spot in the GPI. So that's where I think we are now. And regarding the question about genetic testing for all patients, no, we don't do that. Only if we have a specific reason to suspect for a genetic disorder. And their genetics might help to figure out how likely it is that we get benefit. But in typical cases with no specific reason to suspect genetic dystonias, we don't do testing.